I will be speaking about some of the first embryonic stem cell trials. And as you all know, there are many dozens of human diseases caused by tissue loss or dysfunction. So I will focus today just simply on a few that we are focusing on at Advanced Cell Technology together with our joint venture with Child Biotechnology. And we are pursuing several potential candidates for early clinical translation that involve human embryonic stem cells. And these include retinal degeneration, corneal repair, and then several programs that involve our hemangioblastic-based programs, which include vascular repair, red blood cells, and megakaryocytes to generate platelets. So, of course, the first embryonic stem cell trials are likely to, and indeed are, involving the central nervous system and the eye. And the reason for that is actually threefold. One is they're both immune-privileged sites, so that you can transplant allogeneic cells with an attenuated immune response. But in addition to that, of course, there are major diseases that affect those systems, including Parkinson's, spinal cord injuries, and of course in the eye, visual disorders in the U.S. alone cost the economy over $50 billion, and these of course include macular degeneration. So first I'd like to speak briefly about retinal degenerative diseases. We have recently received FDA approval to carry out two phase 1-2 clinical studies that involve human embryonic stem cells to treat retinal degenerative diseases. And these will entail the differentiation of the embryonic stem cells into retinal pigment epithelial cells to treat both Stargardt's disease, which is one of the leading causes of juvenile blindness, as well as the dry form of age-related macular degeneration, which affects about 90% of people who have macular degeneration. And AMD alone affects over 30 million people worldwide. It's the leading cause of blindness in patients over the age of 55 years old in the US and Europe. And the degeneration of the RPE plays a critical role in the development not only of macular degeneration and Stargardt's disease, but literally dozens of other retinal degenerative diseases such as retinitis pigmentosa. And the transplantation of RPE cells have been shown in animal models to rescue host photoreceptor function. And so we feel that the use of embryonic stem cells are a potentially important and unlimited supply of RPE for transplantation into patients. One of the problems is I'm left-handed and I'm trying to use. Thank you. Okay. So this shows you a cartoon of the eye, and as you can see, the light enters into the eye, and you hit the photoreceptors, the cones and rods that we see with, and underneath those are the retinal pigment epithelium, shown here in purple. And they are attached to the Brooks membrane just underneath these. And the RPE has multiple functions. It's involved with vitamin A metabolism. Here you can see that they're heavily pigmented. This helps avoid the scatter of stray light in the eye. But most importantly, it's critical for maintaining the photoreceptor environment so that they, for instance, the shed photoreceptors are phagocytosed by these cells. And that's critical for the survival of the for the photoreceptors. So there are, again, multiple advantages to using embryonic stem cell-derived tissues for regenerative medicine. First is that you can create a virtually unlimited supply of cells. This is especially important, for instance, for the treatment of these degenerative eye diseases, because right now the main source would be fetuses, and so you can imagine how many RPE cells you can get from the eye of a fetus. So again, the ability to grow up batches of these that can treat literally thousands and thousands of patients is quite desirable. They can be created under GMP conditions, pathogen-free, so we can control every step in the process so that we can eliminate batch-to-batch variation, and they can be thoroughly characterized with extensive release criteria for optimal performance. So we have shown that we can actually create RPE reliably from all the embryonic stem cell lines that we have studied. We've studied literally over 24 different lines that have all reproducibly generated RPE cells. And this is, again, one of the other reasons why people have focused on the central nervous system and the eye, and that is that neural ectoderm is almost a default pathway for embryonic stem cells. So we can get very large, reliable numbers of not only neuronal tissue, but also retinal tissue. So we're able, through this methodology, to create literally billions and billions of cells from very small initial starting quantities. So again, we've studied ES lines that were derived at Advanced Cell Technology. We've studied multiple lines at from the Howard Hughes Harvard, Doug Melton's group, as well as the major lines from Y-Cell. And all of these lines quite reliably generate these RPE cells, and they can be characterized and expanded. So this shows you just what's going on in the Petri dish. And so if you just let your embryonic stem cells overgrow naturally, with enough time, virtually every single culture will eventually develop RPE. If you have embryoid bodies and you leave them in there for months, 100% of those will eventually be covered with pigmented cells. And in a flat Petri dish, this is what you see. You see these little freckles of pigmentation. Those are the RPE colonies, and you can mechanically dissect those out. And one of the features of retinal pigment epithelium is that they de-differentiate and expand through the dish, and then after a period of time, they reestablish their pigmented cobblestone morphology. And they'll go through this de-differentiation differentiation process over and over so that you can start out and create very large numbers of these and then go through that process again and again so that you end up with very large quantities in your final batch. And then we can control the stage that these are frozen down depending on how they work optimally. So you can start out with very lightly pigmented cells. And these colonies are all 99%, almost 100% RPE. And then if you leave them just a little bit longer, they become more and more pigmented until you can actually get very heavily pigmented cells. Of course, the goal here for clinical trials is to be sure that we see the downregulation of the markers of pluripotency and the upregulation of all the various RPE-specific markers. And we have a number of release criteria that involve immunostaining, so we can stain for these various RPE-specific markers, Western blots, RT-PCR. And we see in these batches, they're almost identical to the primary tissue. We see the bestrophin, we see the CREL-BP, we also see the PET-F, RPE65, all again RPE-specific cells. And if you look at the primary fetal cells, you can see that they're almost identical in that regard. And we've also done gene chip analysis, which confirms that they're almost identical. So they look like RPE cells, have all the markers. And one of the main functions is the ability to phagocytose the shed photoreceptor segments. So a quick and dirty way of checking that is you just take some latex beads, you throw them in the Petri dish, and what you see here is that the cells eat these. So that's a very important release criteria that they are actually functional. So everything looks to be fine in the Petri dish, so we wanted to see whether they could really do anything in terms of rescuing vision in animals. So one of the gold standard models is the RCS rat, the Royal College of Surgeons rat, which develops macular degeneration at an accelerated rate, also due to a loss of RPE cells. And what you're actually able to see is here, doesn't want to go down, but basically you start out with a certain amount of visual acuity here in the shams, in the untreated animals, and we can do a dose response. So by adding 5,000 of these cells and increasing it gradually to 50,000, you see a rescue of the photoreceptors. So the visual acuity here is about 100% improved over the untreated controls. And then we see the effect plateaus after a certain amount of cells. So even when you go from 50 to 75 to 100,000, you've pretty much reached your maximal rescue. And we've also studied, there's a mouse model of Stargardt's disease, and what we're measuring here again is visual acuity. And what you're seeing is after about 2 months, you see a drop in the visual function in the untreated animals, whereas with the treated animals, we see normal function. But what I think is most striking is that when you do a cross-section of these treated animals, so this is a cross-section of the retina from one of the RCS rats, and so if the animal is untreated, this layer here, which is the outer nucleated layer, contains the photoreceptors. There's only about one layer of photoreceptors left. That animal is essentially blind. In contrast, the animals that were treated with the RPE, the fresh RPE cells, you can see here very robust 5 to 7 layers thick of these photoreceptors. So again, these are the same cells that we're planning to use in patients. So the hope is that we can see this kind of rescue. So the important thing for the regulatory agencies, of course, is any kind of a product that starts with an embryonic stem cell, the concern is teratomas, tumor formation. And again, the advantage to using these retinal cells is that they grow very robustly, and the process actually is not conducive to undifferentiated ES cells at the end. So we go through this process where we start out with whatever number of ES cells we want. We go through the embryoid body formation and eventually isolate the RPE, and we go through 2 or 3 passages. And we actually, for our clinical product, we're only going to be, in the initial patients, only adding about 50,000 cells into the subretinal space, and that will gradually escalate to a maximum of 200,000 cells. So we developed an assay where we can actually detect one single undifferentiated cell in over 1 million cells. So if we take 10 million RPE cells and throw in 5 undifferentiated ES cells, we can find every single one of those. So that you can imagine, even with our maximal dose, we could detect even a single undifferentiated cell. In fact, we did some in vitro spiking studies where towards the end of the product, we actually added in, we threw in 10% undifferentiated ES cells, and there were virtually none in the final product. And so this is very important, at least on paper. And when we went into the various animals, we also saw, obviously with the undifferentiated cells, tumor formation. This is again in the subretinal space of the eye. But we never saw a single teratoma or tumor in any of the animals we studied. And we even carried out these spiking studies where we added into the product that was going into the eye either 1% undifferentiated cells, 0.1 or 0.01. And again, we didn't see any tumors at all. We saw no safety signals, no ectopic tissue, and we studied literally hundreds of animals. And we followed the NIH 3 mouse, which is extremely immune compromised because, again, this is a discordant xenograft model that we have to follow for the end of the lifespan of the animal, and we indeed want to be sure that we have these human cells in the end to show that they are safe. And so again, we followed them for the lifetime of these animals, and again, we saw nice engraftment into the subretinal space of the cells, and again, everything looked absolutely normal. No pathology was observed. And we also carried out biodistribution studies. The other